Good morning, good afternoon, and good evening, everyone, and welcome to this webinar titled Using Real-World Data to Explore and Demonstrate Comparative Effectiveness. I'm Sydney Menson from the events team here at HealthEconomics.Com, and I'll be your host for today. This webinar has been sponsored by OM1, so big thanks to them for helping to make this webinar possible. We're very fortunate to have Jessica Paulus, Senior Director of Research and Epidemiology at OM1, who will discuss opportunities and challenges with using real-world data in conducting comparative effectiveness research. But just before we get started, here are a few housekeeping notes. First, if the panels on your screen are too big or too small, you can zoom in or out in your internet browser to adjust their size. Next up, the webinar is being recorded, and the video and its associated resources will be available to watch shortly afterwards. We'd love to hear from you and invite you to share any questions, thoughts, or comments in the Ask a Question panel shaking on your screen. We will be taking questions in a Q&A session after the presentation. If you'd like to learn more about what is being presented today, explore the resources panel, where we've added a few relevant links and resources that you can download. And finally, if you happen to have a technical issue, refreshing the page nearly always fixes things, but if it doesn't, reach out to us using the Ask a Question box and we'll help you to troubleshoot. And with that, I'd like to welcome our presenter, Jessica. Hi, Jess. Thanks for joining us today, and the floor is yours whenever you're ready.
Hi, Sydney. Thank you so much for the introduction. And thanks everyone for joining me today and joining Sydney to talk about this topic. So today I'm going to be talking about the major opportunities for generating insights for comparative effectiveness when we are leveraging real-world data. And I'm going to do this while also nodding to some of the unique challenges around using these types of data. So I'm going to start by walking through some of the bigger landscape around methods that are used for comparative effectiveness researches that leverage real-world data. But then I'm also going to go through the lens of a real-world data-based comparative effectiveness study on breast cancer screening mammography that we're doing at OM1. And then I'm going to finish with a few remarks about regulatory grade evidence from real-world data, including regulatory decision-making and some of the special considerations there for comparative effectiveness research. So let's start with just some basic definitions, just to make sure everybody's on the same page. So what are real-world data? They're any data related to patient health status or the delivery of healthcare that are routinely collected. Oftentimes people focus just on the electronic health record data or claims data that are frankly usually the cornerstone of real-world data, but real-world data also comprise more diverse sources, including disease registries, patient-reported data, even insurance underwriting data. We have a really interesting social determinants of health data set that comes from that type of data source. So what are the major aspects that distinguish real-world data from, for instance, trial data? There are three key things that we're going to keep coming back to: the size, the longitudinality, and the representativeness. These three things come together as a really powerful vehicle for comparative effectiveness research. So just a couple more definitions before we really get into it. So this is a framework regarding real-world data-based study designs that was recently published by John Concato. He's of the Center for Drug Evaluation and Research at the FDA. I adapted this a little bit, but it's hopefully giving you a little bit of a sense of what we're really focusing on today. And that's the real-world data-based studies here. Registry-based studies or health claims-based studies. You can see here in this bottom right quadrant that we're talking about studies that make use of observational data, but rest more heavily on the secondary use of clinical data, as compared to the cohort or case control studies that might rely more on primary data collection. Now, of course, we can conduct case control studies, for example, nested within real-world data sources, but I'm drawing a real distinction here between those that rely on secondary data, such as the real-world data, versus those that seek to principally enroll patients prospectively, say, in a hospital-based case control study. I think everyone here is excited about real-world data. At OM1, we're obviously really excited about real-world data, but I just wanted to convince you that this is happening around the world. This is a graph that I made recently from PubMed data showing that interest in real-world data indeed has been growing exponentially over the last 2 decades. This is a PubMed search I did just earlier this month. And you can see here that there are a couple key inflection points. So the first one is the 2009 passage of the American Recovery and Reinvestment Act that created incentives for healthcare technology, including for the use of EHR systems for providers. The concept of meaningful use here is one we all hear about a lot, and it really underscored the importance of not just generating this information, but leveraging it to transform patient outcomes and improve the public health. In other words, to really contribute to a learning healthcare system. And then in 2016 here, you see the 21st Century Cures Act, which was another sentinel moment for real-world data with an emphasis on the modernization of trial designs and the incorporation of real-world data in regulatory decision-making for the FDA. So some really big moments right there. I'm going to come back to touch on clinical trial evidence a bit later in the talk, but I did want to take a moment to emphasize that while trials and real-world data are sometimes presented as being opposing forces, they are but two paths towards a common shared goal. In other words, that's to identify the right treatment for an individual patient at the right time. And whether the goal is to support decision-making for an individual patient or to meet regulatory decision-making goals, these two sources of evidence should be viewed as complementary sources of insights. So let's talk a little bit more about what the opportunities are specifically for comparative effectiveness research insights using real-world evidence. So the first has to do with patient representativeness and heterogeneity. It's been well documented across a range of conditions that trial populations often don't well represent the actual clinical populations that present in clinical practice. Depending on the trial, the question at hand, the trial populations are sometimes at an earlier, sometimes a later stage of disease. And in fact, the trial inclusion-exclusion criteria are designed to do just that, to enrich those study trials with patients who are likely to benefit and unlikely or less likely to be harmed. Now, this is in stark contrast to RWD designs, which typically feature far less stringent eligibility criteria and population winnowing. So this broader range of patients, especially with regards to the baseline risk of patient outcomes, really affords meaningful opportunities to unveil treatment effects, to unveil safety effects that just might not be visible in trials. Number two, efficiency. This is often a really motivating opportunity. So as compared to trials, RWD offer large increases in the number of patients enrolled or available, but also the number of data elements or variables that can be ascertained at relatively reduced resources or costs. So this allows us not just more statistical power, which is great, but also the greater number of data elements can offer potential for profound characterization of cohorts and data elements also for control of bias. Last aspect here is longitudinality. So this refers to the ability to examine treatment and safety effects over time. So there's lots of data out there on sort of the average duration of clinical trials because of the intense resources that are required to maintain such trials over time. RWD designs are really an opportunity here to look at heterogeneity even in treatment effects and safety effects over time. Let's see. So another key RWD advantage is the opportunity to examine the comparative effectiveness of interventions that are just not amenable to trial designs. What you're seeing here is one of my favorite publications. This was published in the BMJ Christmas issue almost 20 years ago, but it's still hilarious and still relevant. The authors here kind of cheekily conducted a systematic review and meta-analysis that sought to address the problem of there being no randomized trial evidence to support whether we know if parachutes are efficacious at all to reduce death from gravitational challenge. So the authors note that parachutes reduce the risk of injury, but their effectiveness has not been proved with randomized trials. So how could we possibly know if they work or not? Their results say that their search strategy did not find any randomized controlled trials of the parachute. I'm sure there's no one here that would be really amenable to enrolling themselves in such a randomized, double-blinded, placebo-controlled trial. So lo and behold, actually, just about 3 years ago, a randomized trial of parachute efficacy was published, to my surprise, again in the BMJ Christmas issue, which is a must read if you're looking for some good science jokes in December. This paper was published by some real leaders in observational data analysis, comparative effectiveness research, and clinical research in general, Bobby Ye and Brahmaji Nalamothu, who was a senior author. Essentially, they conducted a randomized trial. They went out, and anytime they flew for conference travel or personal travel, they sought seatmates to enroll in this randomized trial of the parachute efficaciousness. But lo and behold, the punchline of this paper was that parachutes did not reduce death or major traumatic injury when jumping from aircraft in this first randomized trial of this intervention. So I think there were only a couple of you who voted no, so you were the winners of this poll. And this is why. So the trial was only able to enroll participants on small stationary aircraft on the ground, suggesting cautious extrapolation to high altitude jumps. So this is, poking fun at the primacy of the randomized trial, while also kind of highlighting the appeal of pragmatic and observational designs to address certain kinds of research questions. So I'll just go on to say that furthermore, trials have been historically very poorly positioned to address questions regarding how treatments may be differently accessed or have varying effectiveness across diverse populations, including those defined by protective characteristics like age, sex, race, ethnicity, and others. This table is from a paper that examined the composition of patients enrolled in randomized trials that were cited in the American College of Cardiology, and American Heart Association guidelines for atrial fibrillation, heart failure, and acute coronary syndromes. And we see that even when aggregated across all of these trials, so this is, hundreds of thousands of patients coming together when we look at these systematically, there are very few patients in older age categories and a stark underrepresentation of women. We also see that 80 to 85% of these populations are white. The prior study I mentioned was looking at cardiovascular disease trials, but we see this pattern across most disease conditions, where the participation of patients in certain racial, ethnic groups, age groups, etc., doesn't represent the full spectrum of patients that we see in real-world clinical practice. And this severely constrains our ability to use trial data to investigate subgroups of patients, for example, defined by social determinants of health, sociodemographic characteristics, and more. So I'm guessing that many of you saw this guidance that the FDA published about a little over a month ago on improving the enrollment of participants from underrepresented racial and ethnic groups in trials. In this document, you'll see steps for developing a race, ethnic-based diversity plan. These are highlighted for the importance of applications for new investigational new drug applications, as well as for devices. And there's a focus in here on diversity based on race and ethnicity, but the FDA actually defines a much broader set of underrepresented groups, including by sex, gender identity, age, pregnancy status, and more. So we walked through some of the opportunities that real-world data can afford, and this cartoon brings home some of the caveats of working with RWD and observational data more broadly for comparative effectiveness research. So in today's random medical news from the New England Journal of Panic-Inducing Gobbledygook, we see coffee is associated with, coffee can cause depression in twins, but if we spin the wheel tomorrow, what kind of answers will we get? So one of the questions, the really outstanding question to ask broadly and for any given research question is, can we rely on this source of evidence given the opportunities for systematic error? So I wanted to take a little bit of an advanced poll of our audience members. Confounding bias is one of the first ones that we tend to think about with observational data-based studies and RWD-based studies. So just wanted to talk a little bit more about bias. So the major sources of bias in these designs, as I mentioned, of course, is confounding. There's also selection bias and information bias, or the mismeasurement of information on treatment, confounders, outcomes, etc. And all of these things can lead to spurious findings or can mask or distort associations that truly do exist. And what's been particularly exciting is that as RWD has proliferated, thinking back to that kind of Excel graph I showed with those PubMed hits, in step with the proliferation of RWD has been the proliferation of methods to offer more advanced tools for confounding control and other types of bias in real-world data. Yet some of the things we're learning as we evaluate the kind of impact of some of these tools is that it's often, quote unquote, the basic principles of study design regarding the crafting of real-world data-based research questions that correspond to actionable interventions, the selection of populations, and the recognition and incorporation of patient heterogeneity. These are as important as some of the more fancy, sophisticated, or modern analytical tools for reduction of bias. I'll also add again that the empirical and other types of investigations are exploring exactly what the best method is for addressing confounding in a certain context. Wanted to talk a little bit more about RWD-based comparative effectiveness designs, especially how they perform in comparison to randomized trials. And the importance of basic study design principles came through in this recent RWD-based emulation of a randomized trial that was comparing ablation versus medical therapy for managing patients with symptomatic atrial fibrillation. The Cabana trial you may know about, published in JAMA a few years ago, included a little over 2,000 patients who were randomized to receive either catheter ablation or medical therapy. What's fascinating about this case study is that in parallel, Peter Noseworthy and colleagues, this is on the right-hand side here, conducted an RWD-based emulation of that trial off of the protocol alone that included nearly 200,000 patients. And this was an effort to explore the treatment effect in routine clinical practice. The two publications were timed to be released simultaneously, such that the outcome of the trial wasn't publicly known to the investigators who were doing the RWD-based analysis. So this is to kind of prevent contamination or, kind of p-hacking or other types of activities that the RWD-based investigators might have been biased by. And what was a really interesting moment for those of us who these parallel investigations had a lot of attention from those of us who think a lot about observational data, because what ended up happening is that the results from the RWD-based analyses were broadly similar to those from the trial, which offered reassuring support about the internal validity of the trial results, especially with regards to how well the two interventions how they would be compared in kind of a slightly different patient population in real-world practice, for example. However, I will say the RWD-based study noted that when they did subset analyses for subpopulations that hewed most closely to the trial eligibility criteria, that the treatment effect varied. In particular, the treatment effect for ablation was more modest in the patient groups who would have been excluded from the trial. So this example also brings home how complementary evidence is so important when we're looking at RWD-based CER studies that accompany trial designs. Now, this Cabana study and the RWD comparator were just a kind of N of one evaluation. But I did want to point to many ambitious and large-scale investigations that are ongoing at the moment that seek to compare or evaluate the ability of RWD-based studies to generate estimates of treatment effects, in particular with comparison to randomized trials. So what you're seeing here are two links to two of the major initiatives that I'm aware of, the Operand and Duplicate initiatives. These are actually both partnerships between pharmacoepidemiologists at Harvard and other RWE stakeholders and leaders, including the FDA, Optum Labs, and more. So just to kind of summarize where we are, some of the things we've talked about so far, RWE-based comparative effectiveness studies afford this opportunity to look at treatment effects in populations that are typically often excluded from trials. This can allow us to detect treatment effects that may vary across patients. RWD-based studies also provide giant efficiency gains, larger sample sizes, more extended follow-up. And the trade-offs here are related to what the strengths are from randomized trials or randomized evidence, which is that in a highly structured and supported environment, there's an ideal landscape to minimize bias, in particular confounding bias. So I wanted to transition a bit to our case study today. Our case is based on a leading 3D mammography device company that sought to partner with both key opinion leaders and OM1 to identify and drive best practices in breast cancer screening and treatment. So the goal here was to generate high quality evidence, RWD-based evidence, that could be used to support clinical practice guidelines, to enable rapid claims expansion, and also to inform the direction of a product roadmap. The sponsor here saw the opportunity to do this specifically with RWD, in particular electronic health record data and tumor registry data, to address key questions about the efficacy of their 3D mammography technology as compared to 2D digital mammography. So what was kind of known at the time? Randomized trials had supported the superiority of 3D versus 2D mammography along some important outcomes. But there remained many key evidence gaps that could be best addressed, at least efficiently, with real-world data. Both the U.S. Preventative Services Task Force and radiology and cancer clinical societies had raised the question regarding the stage distribution of detected cancers. This is with the recognition that it's really the long-term reduction in the rate of advanced cancers is really the most important. It's most clinically significant. There's all kinds of issues if the 3D technology was simply surfacing cancers that didn't have a kind of big prognostic impact. And there were other questions that were outstanding regarding the longitudinality, in particular the persistence of benefits for 3D versus 2D mammography with subsequent mammograms. So it's one thing to look at efficacy at the first screen, but what happens at the second, the third, the fourth screen, conditional on prior negative results, for example. And I'll just add that there is an ongoing randomized trial that was designed to address some of these questions. However, the results aren't expected until the year 2030. In addition, that timeline is a bit in question, owing to some recruitment challenges that are probably not surprising to anyone who has tried to do research with primary data collection in the last few years. So that brings us to the study design here. This study draws from a mix of 5 different sites, a mixture of academic and community sites with the goal of recruiting over 1 million women and many, many more mammograms. We at OM1 are collecting and processing the data and conducting the analyses for what comprises a really broad and ambitious research program, including both regulatory grade and research grade CER for FDA submission. So just want to share with you one of the first publications from this RWD-based platform indeed showed that the improved performance with 3D versus 2D mammography was maintained over multiple years and over multiple screening rounds as women came back for annual exams. And at the time of this publication, this was the longest follow-up with cancer registry matching that had been published so far on this question. I've talked a lot about patient heterogeneity and kind of what RWD can offer in terms of thinking about health equity. So I wanted to spend a little bit of time talking about that here. A key objective for this program is leveraging the opportunity to build a more diverse cohort, in particular to explore the efficacy of 3D versus 2D mammography in underrepresented and high-risk subgroups, including Black and Asian women. So we know that breast cancer carries one of the highest observed racial disparities in mortality. This is a complex problem attributable likely in part to barriers in access to screening. It's been observed that Black women tend to have more aggressive breast cancer subtypes at an earlier age as well. But in fact, the sponsor of this program has a deep commitment to health equity and women's health in particular. And this RWE-based research program is one of the key ways they're delivering on that commitment. So the cohort, the RWD-based cohort, features a greater proportion of Black women than the US population at large, and of course also a dramatically higher representation than corresponding clinical trials of breast cancer screening modalities. So this is a recent publication from the Journal of the American College of Radiology. Using this RWE platform, we were able to demonstrate that Black women, as expected, have reduced access to cutting-edge 3D mammogram technology. However, we also were able to look at whether the comparative effectiveness of 3D versus 2D mammography was different according to a patient's race ethnicity. So the take-home message here was that Black women do indeed have reduced access to 3D technology, but they are not less likely. They're not less likely to benefit from the improved screening profile of 3D mammography than white women. So I wanted to take a little bit of time to transition to think about regulatory decision-making and how this all fits in here. In 2021 was a busy year for the FDA with regards to RWE. In 2018, the FDA published this framework for their RWE program, but 2021 brought the arrival of 4 additional guidance documents for industry that offered much greater detail on standards for RWD from electronic health records and claims and registry data, as well as a guidance document on data standards, as well as considerations for design. I will say that these guidance documents don't have any content on study design and analysis of real-world data. So some of the questions that we have talked about so far, what's better, instrumental variables, propensity scores, how should we make the propensity score, how should we apply the propensity score? These guidance documents are essentially silent on these issues. However, there are additional guidance statements that are forthcoming that are going to offer some insight on some of these questions that often we spend a lot of time thinking about in protocol development and statistical analysis planning. So our teams, as well as industry alliance groups, have combed through these 4 guidance documents very carefully. And I wanted to share with you what I think are the cliff notes. There are 3 main points of relevance in particular for comparative effectiveness research studies. The first one on the left-hand side is transparency. So this is the recommendation that protocols and statistical analysis plans are submitted for agency review, for FDA review, in advance of study analysis. The second concept in the middle has to do with relevance, or in other words, that means whether data are fit for use, quote unquote. What do we mean by fit for use? This refers to whether the data that are collected are actually corresponding to the appropriate patients for inference, and whether the necessary data elements are available. So for example, these guidance documents are really asking for a quite detailed a priori plan and justification for which sites or health systems or data systems are being sampled for the study, why the included patient or catchment populations are likely to be the appropriate patients that you're going after, and that the data elements are available from all of those sources, whether it's structured, unstructured data, et cetera, that will adequately characterize the severity, outcomes, just offer profound enough characterization. When these protocols are reviewed, this fitness for use is often the major subject of attention. I would say more so than consuming time regarding some of the details around statistical analysis. But there's a huge emphasis on who are the patients, why have they been selected, why are they appropriate for the research question, and is there enough data, the adequate data that can offer characterization. especially with regards to the patients that might be in randomized trials and or in real clinical practice. The last principle here on the right-hand side is reliability. And this has to do with whether the data are accurate, complete, and traceable. In other words, this has a lot to do with misclassification bias that I mentioned some moments ago. And one of the real key words here that came out of these guidance documents was validation. So the FDA is really asking for validation of the priority data elements. In particular, those you should start thinking about the treatment or the exposure, the outcome, as well as key confounders. And what the agency is really asking here is to validate some of these data elements with another source of information, ideally a gold standard. This is oftentimes a very big ask when we're looking in real world data. It's not uncommon for us to really only have one source of data on a particular data element. We might, for example, be combing a radiology note or a pathology note for the particular cell type of cancer. And that might be the only place we expected it to show up, which doesn't really leave any room for there even being a gold standard if there's no kind of second source of information. And I think the FDA recognizes that the kinds of gold standards that we often think about for other research settings are simply not possible with this type of evidence. Nonetheless, they really challenge us to, I think, be creative, come up with proxies, even if it's not necessarily a validation, but a corroboration between other sources of this variable or data element that are coming from other pieces of the medical record, for example. So at OM1, we've had to develop some of these validation plans for RWD-based studies going to the FDA and have had to be very creative and resourceful in trying to identify other sources of quote-unquote validation or corroboration. So I wanted to share just a couple resources before I conclude. The first one is that for anyone who is interested in a primer on best practices for the conduct of RWE-based CER studies, the two professional societies that are most in the sphere, ISPOR and ISPE, assembled a joint task force to generate this set of best practices. If you read this article, I think it might be linked here, this should remind you of the epidemiology courses that you might have taken in your training, if you were lucky enough to take epidemiology courses. As a lot of this really just comes down to general good study design, which has been kind of a theme in my talk. You'll see the same themes in this task force guidance as come up in the RDA guidance, the FDA guidance documents on RWE. A lot of this is simply about good common sense study design as well as transparency. So on that note of transparency, one of the salient themes around some of these studies is the notion of building credibility and trust, in particular around analyses that might be pre-specified versus might be post hoc. This is to address concerns of data dredging or key hacking, etc. So these are generally concerns that I think affect all study designs, whether we're talking about RWD or not. But until now, to my knowledge, there had not been a dedicated place to register your RWE-based study protocols a priori. A consortium of organizations that you can see here with the icons, the NPC, ISPOR, the Duke Margolis Center, and ISPE, recently launched the site where you can publish your protocols for RWE studies, CER studies, or other studies. Many of us were using clinicaltrials.gov to do this, but I'm sure many of you also felt a little odd putting your observational study on a website called clinicaltrials.gov. So I encourage you to poke around here. As I mentioned, this is recently launched, so you won't see thousands of protocols here yet. But this is a great place to store your protocols, and in particular to tell the FDA that you have stored your protocols here in a public-facing place a priori before the collection of data or before the conduct of analyses. So I wanted to take some moments just to reflect on some conclusions from what we've talked about today, and then we'll transition to some questions. So first is that RWD, as I've mentioned, can catalyze insights really in comparative effectiveness. And it does this through the unique cocktail of opportunities that are afforded by these data sources. We see a much broader range of patients with regards to demographic characteristics, of course, but also with regards to something critically important for treatment effects, and that is the risk of the outcome. So in randomized trials, you see oftentimes a certain homogeneity of the risk of the outcome that comes through the imposition of the eligibility criteria. In RWD, that's not the case. So we will see more often patient populations that have the full spectrum of risk of the primary outcome of the investigation. And that can be a very statistically and clinically powerful asset. We also see things we would expect, like larger sample sizes, extended follow-up, and we see opportunities to really profoundly characterize some cohorts in ways that we might not, frankly, with other types of investigations. So one example of this is that I'm doing an RWD-based study right now for comparative effectiveness, where we have access to patients' home ownership status. So whether they are a homeowner or not, whether they're a renter and their probability of home ownership, as well as credit risk scores, household income, and more. And it turns out that when we think about disparities, health access to certain health technologies or basic medical care, we see that these variables, these socioeconomic, sociodemographic variables, are often important to explain different types of access issues and even treatment effects. So again, these approaches can really offer a richer data landscape and characterization of populations. What is the elephant in the room? That's the bias that comes from these designs. Earlier, we conducted a poll about what the quote-unquote best method is for addressing some of these biases. In particular, confounding bias has a primacy here, but also misclassification is a huge issue. So whether we can really trust some of the elements that we're getting from real-world data. There are just stunningly sophisticated tools and increasingly sophisticated tools to reckon with some of these biases, both with analytical techniques as well as sensitivity analyses that are really important to do to really kind of kick the tires on whether observations from RWD-based CER studies could be explained partially or completely by unmeasured or residual confounding, for example. But we've also seen through some of the empirical work that I nodded to earlier that some of the ways in which RWD-based studies can really go off the rails is not because you haven't used the propensity score in the most perfect way, but rather because the question that's being asked doesn't correspond to that clinically relevant question that might be the subject of a trial design. So really thinking about having intervention-oriented questions for your real-world data that correspond to real-world clinical decisions. And I put all of this under the bucket of kind of basic study design, making sure that you have the patients through the design of inclusion-exclusion criteria that correspond to trial designs. If your goal is to emulate, your goal may not be to emulate a randomized trial. And lastly, just to point out that the landscape for regulatory decision-making is advancing really rapidly. I pointed to those 4 guidance documents that came out in 2021. The one about developing a race, ethnicity, diversity plan for randomized trials just came out a couple months ago, and we're expecting many more in the coming, I would say, 12 months or so, that are going to give us some more benchmarks, in particular for some of the designs and analyses. So with that, I wanted to just thank you for your attention and take a moment to see if there are any questions.
Thanks, Jess. That was awesome. I'm going to give you just a second to take a breath and maybe a sip of water. And I just want to remind everyone that if you have a question, you can submit it using the Ask a Question box that's located here. We may not have time to cover everything during the live session, but please submit your questions and we'll get to as many as we can. Okay, Jess, welcome back. Ready to dive right into our Q&A. Okay.
Yes.
Our first question here, in your view, are there any disease or condition areas or other settings where an RWD-based CER study just won't work?
You know, that's an interesting question. And I'm scanning down other questions, a couple other questions that I think are related to this. There's a question here about how to deal with rare diseases and small sample sizes that I think I'll try to incorporate. I'm going to try to batch some of these questions together. So are there cases where this just won't work? How do we deal with rare diseases? We could maybe even start there. What I have seen with regards to rare diseases is that RWD is really the best place to start because you're going to get more patients for these conditions than you will through any other approach, especially when you can look over multiple large health systems. I'll give you an example. We're working on one right now for an extraordinarily rare hematologic condition. And yet, when we use real world data, we can assemble a cohort of patients that is larger than has ever been published or that we know of for this condition. And yet, there still may not be enough patients there to accommodate the most complicated statistical analyses, for example. So we know that we need to have observed a certain number of outcomes. We need to have a certain amount of data there to be able to design analyses, in particular when we're talking about controlling for confounding. So while we may have the biggest cohort ever of patients with this rare hematologic condition, we still may be constrained analytically in, for example, how many variables we can adjust for. This is where, again, propensity score approaches really can come to the rescue here because they reduce the dimensionality of some of these issues. So I guess I would say that RWD, even when there may not be an ideal number of patients to support certain kinds of analyses, it can become a really important hypothesis generating exercise. And I will say that based on our experience with regulatory decision making in the setting is that the FDA has had a lot of grace or tolerance for the fact that the sample sizes may not be that which were in your wildest dreams, but that offer a really important glimpse into especially what the real world experience of these patients are, in particular if we're trying to compare the experience of these patients to a much smaller number of patients who might be enrolled in a clinical trial. Another really important question that relates to all of this is the availability of certain confounders. This was a question from one of the attendees, and I would say that this is less negotiable. So with regards in particular, if you're interested in conducting these studies simply for research insights, that's one thing where you may decide to go ahead even if you don't have information on the perfect confounder. You may only have information on proxies for the confounder. In our experience, we've seen low tolerance for data that's not quote unquote fit for use. So, there are frankly some ways in which we need to characterize prognosis that are poorly documented in real world data. I think there's sometimes a little bit of over optimism about what your clinicians might be doing when they write the notes after your visits. Um, even for some conditions that are, prognostically very serious. Um, it's not uncommon for us to go look for information on severity, um, and to see that, in a given medical subspecialty, there's just very low documentation of risk scores, for example, other types of prognostic indices that randomized trials will compel you to calculate or provide the data elements to do. So this comes back to kind of the real importance of thoughtfulness in study design. And this comes back to the FDA's strong recommendation that you run protocols and statistical analysis plans by them before you proceed with the research. We've found that that has offered really important clues about what variables the FDA thinks are most important for making sure you have validation plans, for example, for certain variables and not others. Of course, there are, clinical experts that are really important to consult here to kind of come up with a wish list of confounders. But that feasibility assessment essentially should be part of the design of the study before it ever launches. So what is the conceptual definition of the variable you need? What is the operational definition of how you're going to extract it from real world data? And if some of that is a no-go, are there proxies that are available? How good are the proxies? How well do they perform at coming up with a sense of that variable, even if it is frankly missing? In our work, we do a fair bit of estimation and imputation when these variables are more missing in real world data than you would prefer. One example is PHQ-9 scores, or kind of that depression severity index that your primary care doctor and other providers may ask you to do when you come in for an annual physical. We see that observed in real world data, but using available data, we're able to estimate it even for patients that don't have it necessarily documented by their healthcare provider in the EHR. So that's just to say that you've surfaced some really important aspects to consider when you're deciding whether to embark on this adventure, but there are also a lot of creative solutions, both to deal with missingness and also just, opportunities for assembling really clinically meaningful inferences, even when the disease is rare.
Great. That was a really awesome detailed response. Our next question here asks, what are the most important aspects to consider when designing a regulatory grade CER study versus a research grade?
Yeah, so this question is related a little bit to what we talked about before. You will find, so I think it goes back to one of those first principles is to get the smartest, best methods people and clinical experts together to really do a feasibility exercise. That's both, as you're developing the protocol, I mean. So when you think about the health systems you're enrolling, or the sites or the data sources, wherever this data is coming from, is to really think about the correspondence or the fitness of those patients, those populations, and their relevance to the research question of interest. You will see a lot of interrogation of that particular point. So are you collecting sources of patients that are suitable? And then coming back to the same theme, is the data available that is going to actually allow you to answer this research question validly? Because if you don't have the key confounders, you're going to get a biased answer that is not going to be useful. So a lot of this comes down to making sure that there is feasibility assessments done, data biopsies. It's worth investing somewhat in actually working with different data sources or health systems to make sure that the data elements that they tell you are in there are actually in there. We've talked about optimism a little bit, but we find that when you ask physicians sometimes, even key opinion leaders within practices, if a certain variable is going to be in the EHR, they are sometimes surprised to learn that it's not well documented across the health system, that there's heterogeneity in providers, that there's heterogeneity across practices. So we find that really doing some pilot investigation work with it's often with the IT folks at some of these health systems who are able to work with us to actually assess how often is the PHQ-9 really in there, even if Dr. X says that she does it all the time with her patients. And lastly, just transparency. So registering that protocol somewhere and submitting it for review by the FDA, assuming it's regulatory grade. I would say that even if it's not regulatory grade, putting it on that RWE registry-based website that I showed you a couple slides ago is a really good practice. You see increasingly journals that are forcing even observational studies to have a protocol that is publicly available, quote unquote, or that it's been pre-registered somewhere. And so that RWE registry is a good place to do that. And the last thing that is just critical for the regulatory studies is the validation, which is an extremely challenging problem, to be honest, just given the gaps in real world data that are not, they're nobody's fault. It just is what it is. But sometimes folks, especially who might have spent decades only thinking about clinical trials, are unpleasantly surprised by the fact that, for example, getting breast cancer stage data, even from a gold star cancer registry, is just not as prevalent as you would hope. So for some cancers, even using creme de la creme cancer registries, we see 40% missingness on stage. In some cases, it's much higher. We see, much higher prevalence of these data, but that's really where a lot of this work needs to happen, is coping with the missingness in a way that is satisfactory.
Great. Awesome. Thank you. I think we have time for one or two more. So let's jump right to our next one. Has the FDA to date incorporated a study being listed on the new ISPOR ISPE Duke Margolis RWE registry in the agency's review process of a particular product? It's a little bit long-winded.
Yeah, I'm just reading this question again. This question is an interesting one, but I think we don't know yet because the RWE registry is so recently hatched. I think that I found out about it just about 2 months ago. And I think I found out about it just shortly after it was launched. So I think not enough time has elapsed before we can answer your question. In particular, what reviews have been completed. But I think I will certainly be really curious to know the answer to that question.
Yeah, that makes a lot of sense. Okay, let's do this one. How do you deal with medical specialties that are primarily office-based and use a sleuth of EMR/EHR solutions and are not consolidated or integrated?
Yeah, this is a really good question, a really thorny question. So we often struggle with this, even today, even in health systems that use, some of the more, the top two, for example, EMR, EHR based systems. In particular, we struggle with encounters that patients may have outside of an office where they are receiving their care. Maybe they're going someplace else for labs, maybe they're going someplace else for imaging, and sometimes these reports are shared with kind of the primary office, to your question, but they come through in formats that are difficult to deal with. They come through in images. So for example, a PDF of imaging report or a lab report, they come through, and other things that are just less wieldy to deal with as compared to structured data. That is the way that we recommend. It becomes a more effortful process, to be clear, in particular because you need to plan resources to process those quote unquote unstructured data sources. And even upstream of that, you need to make sure you have those data sources. So this reminds me a little bit of the conversation we just had about missingness, but I'd say that there's two key aspects here. There's one, doing more heavy lifting than we would expect in an RWD based study here, chasing down some of those other reports, for example, making sure there's the resources to do that, making sure that you have the adequate technical expertise to do approaches to kind of process the unstructured data using natural language processing, especially if it's high volume, machine learning, et cetera, to deal with some of those reports that are coming through from other sources that might not be in the format of your primary office data. And then the last thing is just the missingness. So it's accepting, making peace with the fact that this is going to happen, and making sure you have an analysis plan that addresses the size of the threat of the bias, tries to reduce it, and then tries to anticipate impacts on the study. So I think it's one thing to say, oh shoot, we have a lot of missingness, and that's not going to go over well with anyone. But the more sophisticated thing is to quantify the amount of missingness, to try to reduce it, for example, through an imputation approach, but then to anticipate the direction of the bias, the direction and the magnitude of the bias, and to do analyses about whether this bias could be completely, for example, explaining the result that you see, or might be explaining only a part of it. In other words, that there's a signal that it still remains, that is kind of valid and not likely to be explained by missingness.